So I also suggested that you take a look at the NowSolveThis 14-2. So let's take a look at that together here. Now this is another biosynthetic pathway problem using genetics to elucidate a biosynthetic pathway and discover all the genes required to make these important biochemicals. So here is one involved with the production of tryptophan in a bacterium called Salmonella typhimurium. So a series of mutations in this bacterium results in the requirement of either tryptophan or some related molecule in order for growth to occur. So we have the mutant and we need to add tryptophan in order for the bacterium to grow, or a related molecule. From the data shown here, suggest a biosynthetic pathway for tryptophan. Okay, so let me see if I can paste this here. Copy. Ooh. Okay, so here's our data. We have 4 different mutants here: TRP8, TRP2, TRP3, and TRP1. And again, I like to sketch these out, and I encourage you to do the same. So TRP8, 2, 3, and 1. Do they grow on minimal medium? None of them do. Okay. Do they all grow when we add tryptophan? Yes, they do. Okay, so they're TRP mutants. Now, why are they TRP mutants? They're probably missing some enzyme required to make tryptophan, and it doesn't go from a simple precursor straight to tryptophan. There are a bunch of intermediate molecules that the bacterium here needs to make. And so we take a bunch of molecules that we think are along this biosynthetic pathway, and here we have anthralic acid, indole glycerol phosphate, and indole. Now anthralic acid, if we add anthralic acid, TRP8 can grow. So we're adding anthralic acid to minimal medium, and minimal medium again is just a medium with simple carbon source, simple nitrogen, minerals, and H2O. And the normal wild type bacterium can grow on this. But for a TRP8 mutant, we need to add anthralic acid, IgP, or indole, or tryptophan. So we add any of these and the TRP8 mutant can grow. For TRP2, adding anthralic acid does nothing. It still won't grow. But if we add any of the others, it grows. TRP3, anthralic acid or indole glycerol phosphate do nothing. So we can add those to the medium. It won't grow. But if we add indole, okay, it can grow. And TRP1, none of these. So we can't add anything. We can't get it to grow unless we give it tryptophan directly. So what we need to do now is propose a biosynthetic pathway for tryptophan, and place these different mutations, you know, or at least the genes for the enzymes in that pathway at the appropriate position along the pathway. So I think this one, let's hope this one is straightforward. Okay, this is one of the NowSolveThis problems, and those are usually very straightforward. So let's start with our pathway with tryptophan at the end of the pathway. And let's see, what do we want to put next? I'm going to go with indole. Let's just go in order. The same order that they're listed in the table. So here's our proposed biosynthetic pathway, and I'm going to put simple precursors over here. Simple precursors, so just simple carbon compounds somewhere. They have to start somewhere, and they start being built up into more complex molecules using enzymes that the organism's genes encode. So here's one possibility. So we go from a simple precursor to anthralic acid to IGP to indole to tryptophan. And what are the genes or the enzymes involved in this process? So the TRP1 mutant. The TRP1 mutant will only grow if we give it tryptophan. So I'm going to guess TRP1 encodes an enzyme that's also called TRP1. So the gene names and the protein names are usually the same. So TRP1 functions right here. So the TRP1 gene encodes the TRP1 enzyme, and that functions right here. TRP1 converts indole to tryptophan. And if we look at our data, that seems to be completely consistent with all of the data, right? So TRP1 mutant does not grow if we give the bacterium any of these compounds here. So if we give it anthralic acid, does it grow? Well, no, because if TRP1's mutated up here, its anthralic acid is just going to be converted to this, which is going to be converted to that, which is going to be stuck because we don't have TRP1. And the bacterium needs this one to grow, to live. Probably doesn't need these. It needs this one because we can give it tryptophan and it lives. So these are just intermediate chemicals that are slowly working their way up to becoming tryptophan. Okay, so far our pathway looks good. TRP1 goes right there. Okay, so what about TRP3? TRP3, let's just put this one right here and see if this is consistent with the data. So I'm proposing TRP3 is a gene that makes an enzyme called TRP3 that converts IGP to indole, and then TRP1 will convert indole to tryptophan. Okay, is this consistent with the data? So TRP3, a TRP3 mutant, a bacterial strain that is mutated in TRP3 cannot convert IGP to indole. So if we give the TRP3 mutant indole, if we provide it with this compound right here, well, then it should be able to grow, and we can see that it does grow. So look at the table up here. It's a plus. Okay. So if TRP3 here is really in charge of converting IGP to indole, it shouldn't grow if we give it more IGP. Because this is the problem. It can't convert IGP to indole. And is that the case? Yes, that is the case. Because look, it doesn't grow when we give it indole glycerol phosphate. Okay. So far, our predictions are totally consistent, completely consistent with the data. Let's take a look at TRP2. I'm going to put TRP2 right here. And so, again, this is a NowSolveThis, so nothing's out of order and everything's making sense. If we just, go from right to left here, everything seems to be making sense. Okay, so TRP2 I am proposing encodes a gene which makes an enzyme called TRP2, and that enzyme converts anthralic acid. I'm sorry, I've been saying that incorrectly. Anthranylic acid. It doesn't really matter. Anthranylic acid. You don't have to memorize that, anyways. So anthranylic acid is converted to IGP by TRP2. That's my hypothesis. So is this consistent with the data? So if this is true, if we provide the TRP2 mutant with anthranylic acid, it should not grow because it can't convert it to IGP. Is that the case? This is the case. You can see whoever did this experiment added anthranylic acid to the culture medium that had the bacterium TRP2 mutant, and it did not grow. However, adding anything else does work, right? And that makes sense. So if TRP2 is mutated here, this step is blocked. If we give the bacterium IGP, or we give it indole, or we give it tryptophan, Well, let's just say we give it IgP. If we give it IgP, well then IgP, that bacterium still makes TRP3. TRP3 is fine, so we can convert, TRP3 can convert IgP to indole, and TRP1 can convert indole to tryptophan. So we can give the TRP2 mutant IgP, we can give it indole, we can give it tryptophan. Any of those will allow it to grow because it is blocked at this step. We can't give it anything down here. We can't give it anthranilic acid or any other compound. It won't help it grow. We have to give it something downstream of the TRP2 step in the biosynthetic pathway. Okay, so far all of our data are consistent, but what about TRP8? We don't know where TRP8 goes, so I'm going to guess TRP8 must go down here somewhere. So TRP8 is needed to go from some simple precursor to anthranilic acid, or possibly an earlier step to a simple precursor to a slightly more complicated precursor, which is then converted to anthranilic acid. So TRP8 is required before the formation of anthranilic acid. So it makes an enzyme called TRP8 that converts some chemical to anthranilic acid directly or a precursor to anthranilic acid. And how do we know that? Because if we give it anthranilic acid, well then we bypassed whatever step was blocked because TRP8 is mutated, and then anthranilic acid can be converted to IgP by TRP2, which can be converted to indole by TRP3, which can be converted to tryptophan by TRP1 in the TRP8 mutant, because only TRP8 is mutated. Okay, and the same thing, we can give it any of these compounds in the bacterium, the TRP8 mutants will grow. Okay, so we have a pretty good model here of the tryptophan biosynthetic pathway in this bacterium, and we have 4 different genes which make 4 different enzymes involved in the process, and it looks like we probably placed them at the correct locations in the biosynthetic pathway.